Hello everyone, welcome back to my video tutorial for single-cell RNA sequencing data analysis. Today I'm going to show you how to use Azimuth reference data to annotate your single-cell RNA sequencing dataset. Azimuth reference datasets are very useful if you are new to single-cell RNA sequencing data analysis, especially you don't know well the marked genes for the cell types in your dataset, then Azimuth reference mapping is a very useful method for you to start your data analysis. So Azimuth is a website aims to build all the reference dataset for human tissue. So at the moment they have 14 reference dataset. You can see down from here they have the human PBMC, human and mouse motor cortex, pancreas, fetal development, kidney, bone marrow, human lung, tonsil, adipose, heart and the liver. They also have two reference datasets for the single-cell ataxic data analysis. You can go to the app to upload your dataset, but today I'm going to show you how to map in your dataset using Azimuth reference in R. If you watched all my videos, most of my demonstration I'm using the non-single-cell RNA sequencing data, so we can download the human lung reference dataset and use it to map other dataset in R. Let's click this link. You can see here is the human lung V2 reference dataset. If we go to the Xenode website, we can download the reference dataset for the human lung. You need to download both files, IDX and reference RDS. So I downloaded both files already. If you have a look in my file, in the reference folder, I have the IDX file and the reference RDS file. For today's demonstration, I downloaded the dataset from the GSE 136831 dataset. So now we can go to RStudio and do the analysis. So first we need to load the packages Surat, Azimuth and Tidyverse. So we can read the reference data in. You can see in the data window we have a Surat object named as non-reference. So let's have a look at the metadata for the non-reference dataset. So you can see from the reference dataset we have 584,884 cells in this dataset, and in the metadata column you can see the cells were annotated at a different level. You can see level 1, level 2, 3, 4, 5 and the find level. At level 1, they just labeled the cells as immune cells, epithelial cells, stroma cells, and endothelial cells. At a higher level, the cells were annotated in more details. So we can use the dimplot function to have a look at the cell annotation at level 1. So let's run the dimplot. Okay, you can see at level 1, cells were annotated the epithelial cells, endothelial cells, stroma cells, and immune cells. We can also plot the cells at the finest level. So let's plot the cells. So let's zoom in. You can see at the finest level, and you can see from here the annotation for each cell cluster. If we have looked at the Azimuth website, you can see our reference plot show the same shape of all the cell clusters, which means we downloaded the correct reference dataset for our analysis. So now we can read the GSE 136831 dataset in as the non-query dataset. You can see we have a Naji Surat dataset named as non-query. If we have a look at the metadata for the query dataset, you can see we have 96,303 cells in this dataset. In the metadata column, we have the origin ident as the Surat project. We have the uncount RNA, unfeatured RNA, cell ID, and the patients are control. And in this dataset for demonstration, I only included the cells from healthy lung. So we noted the reference data and also the query data. So we checked the reference data. It is the correct reference data set for the cell types in human lung. And also we noted in a query data set. So now we can use the run Azimuth function in Surat to annotate our non-query dataset using the reference dataset. So the Surat object will be non-query and reference data file directory is in my reference file. We know there are two files in the reference file. One is the IDX file and the other one is the reference file. So let's run Azimuth annotation for the query dataset. So it will take some time to run the mapping and the annotation for the query dataset. Okay, so I finished the mapping and the annotation for our query dataset. So now we can have a look at the metadata for the query dataset again. So you can see now in the metadata query dataset, we have the percentage mitochondria DNA predicted score for each level and also the cell types for each cell at the different levels, level 1, level 2, 3, 4, 5, and the finest level. So now we can use the annotation to plot the cells for the query dataset. We can plot the cells at At level 1, we know we have 4 types of cells at level 1. You can see our query dataset. The cell cluster shape is the same as the reference dataset. We have epithelial cells on the left side. We have endothelial cells on the top, stroma cells in the middle, and the immune cells are here. If we plot the cells at the finest level, we should also have cell clusters. So let's zoom in. You can see the cells were annotated at the finest level as the reference dataset. So now let's have a look at the cell ident for the non-query dataset. If we run the idents for the query dataset, you can see at the moment the ident for each cell is the origin ident surrogate object. If we simply use the dimpnol and don't group the cells by different level, you will see all the cells will be labeled as surrogate object. So you can set the cell idents at a different level. For example, we set a cell ident at the finest level. Let's set it. Then we plot the cells again. And we zoom in. You can see now the cells were labeled by the finest level without the group by function. If we look at the cell ident again, you can see each cell was labeled as the cell types at the finest level. So we use the Azimuth reference non-dataset and annotated our query dataset. So now we can perform more analysis. For example, we can use the feature plot to show the marked genes for different cell clusters. Before we do more analysis, we need to normalize the query dataset again. Because when we run the Azimuth function, Azimuth normalizes the data before mapping, but Azimuth does not return the result for the data normalization. So we need to normalize the data again. Then we can use the feature plot to see gene expression in different cell types. For example, we can plot the EPCAM is the marked gene for epithelial cells, PDPRC is the marked gene for immune cells, CLDN5 is the marked gene for endothelial cells, and the collagen 1A2 is the marked gene for mesenchymal cells. So let's run the feature plot function for those 4 genes. Let's zoom in. You can see now we can visualize gene expression after Azimuth mapping and annotation. So we use the feature plot as the example. You can do more analysis for your data set. And also you can save your annotated data set for future analysis. Okay, so I finished today's demonstration. In summary, I showed you how to use Azimuth reference dataset to map and annotate your own single-cell RNA sequencing dataset. I hope you can see from my demonstration, the Azimuth mapping method is a straightforward method for your data analysis. I'm going to stop here for today's demonstration. Please subscribe to my channel if you haven't to do so, and share my videos with your friends. Thank you, and I hope see you next time.